Thank you to the organizers for the opportunity to present our study at the ANZSNM annual conference. The project I'm presenting today is titled In vitro and in vivo characterization of zirconium-labeled anti-VISTA antibody CI-8993. This study is a collaboration with Curis. Curis is a biotechnology company focusing on the development of first-in-class and innovative therapeutics for the treatment of cancer. With this slide, I would like to disclose that this project was funded by Curis. CI-8993 is a fully human IgG1 kappa monoclonal antibody that binds specifically to immune checkpoint molecule VISTA. VISTA stands for V-domain Ig suppressor of T-cell activation. Interestingly, murine anti-VISTA antibodies can overcome anti-PD-1 and anti-CTLA-4 resistance in preclinical tumor models, and this antibody is currently being developed as a combination therapy for negative checkpoint regulator blockade. So how does this work? Most of you are probably familiar with the negative checkpoint regulators expressed on activated T cells, CTLA-4 and PD-1. Once bound to their ligands found on the tumor cell, these receptors reduce T cell responses to effectively attack tumors. Currently, blocking antibodies against these molecules have shown durable responses in multiple cancer patients. However, not all cancer patients show effective responses to this form of therapy. Unlike all other negative checkpoint regulators that are expressed on activated T lymphocytes, VISTA is expressed on naive T cells and thought to be a negative regulator of T cell activation. In multiple murine cancer models, VISTA has been found at high expression levels on myeloid cells that infiltrate the tumors. When expressed, VISTA keeps the T cells in a quiescent dormant state by blunting the production of T cell cytokines and activation markers, and this is thought to be the mechanism rendering tumors resistant to anti-PD-1 or CTLA-4 targeted therapy. The aim of the study was to develop zirconium-labeled CI-8993 for PET imaging and quantitation, and to validate zirconium-labeled CI-8993 in preclinical tumor models prior to a planned human trial. Two antibodies were used for chelation. The first one is CI-8993 investigational drug product that binds VISTA, and the second one is an IgG1 isotype control antibody that does not recognize VISTA and has no targets expressed in tumor or other normal mouse tissues. CI-8993 and isotype control were chelated with the bifunctional metal ion chelator desferoxamine at a 5-fold molar axis using the method previously described by Vossian and colleagues with some modifications. Conjugation was performed in sodium bicarbonate buffer at room temperature for 1 hour. For purification, PD-10 gel filtration and a sodium succinate buffer were used. This procedure resulted in stable constructs with an average chelator to antibody ratio of 1.81. SDS page showed that integrity of chelated CI-8993 and IgG control was maintained. The reaction time for radiolabeling was 15 minutes at room temperature, and again for the purification of radiolabeled antibodies, we used PD-10 gel filtration. The radiochemical properties of the radiolabeled CI-8993 are shown in this table. The specific activity varied between 182 and 220 megabecquerel per mg. Radiochemical purity was assessed with instant thin layer chromatography, and antibody integrity after radiolabeling was checked with size exclusion HPLC shown in the figure on the left. After 7 days of incubation in human serum at 37 degrees, the radiochemical purity was still 88%. To test the human VISTA binding activity of radiolabeled constructs, we used an ELISA assay using recombinant human VISTA antigen. This assay demonstrated that binding activity to human VISTA was maintained after radiolabeling. For the imaging studies, 1 mg per kilo radiolabeled CI-8993 or isotype control was used, and the amount of injected radioactivity was 124.7 microcurie for CI-8993 and 152.7 microcurie for the IgG1 control. In a first PET imaging and biodistribution study, we evaluated the tissue biodistribution of zirconium-labeled CI-8993 in a syngeneic MB49 bladder cancer tumor model. MB49 tumor cells carry the male HY antigen, and therefore these tumors are immunogenic in female mice and can trigger T-cell immune responses. We injected MB49 tumor cells in human VISTA knock-in female mice or wild-type BLAK6 control mice. In wild-type mice, myeloid cells including T cells will express murine VISTA. The human VISTA knock-in mice carry a chimeric version of VISTA protein, and in these mice, myeloid cells carry the extracellular domain of the human VISTA protein linked to the murine intracellular VISTA protein domain. Therefore, the extracellular part of this molecule will be recognized by CI-8993 antibody, and the intracellular part will still be able to signal and regulate immune responses. Images shown are representative whole body surface rendered MR images, maximum intensity projection PET images, and fused PET MRI images. MB49 tumor-bearing human VISTA knock-in mice or wild-type mice were imaged on day 3 post-injection. Shown in the top row on the left is a mouse injected with 1 mg per kilo zirconium-labeled CI-8993, and on the right, radio-labeled CI-8993 was co-injected with 30 mg per kilo of unlabeled CI-8993. The red arrows indicate location of the tumor. We also imaged these 2 different doses in wild-type BLAK6 mice shown in the second row, and at the bottom we show the images of radio-labeled IgG control antibody injected in a human VISTA mouse shown on the left, and radio-labeled IgG control antibody injected in a wild-type control mouse shown on the right. In the human VISTA mice, at the 1 mg per kilo dose, high uptake was seen in organs containing myeloid cells, such as spleen and bone, but at this dose level, no uptake was seen in the tumor. In the presence of 30 mg per kilo unlabeled antibody, uptake in spleen and bone tissue were reduced, and as a result, the tumor uptake was increased. Comparing these images to the images of the wild-type mice, we see a stark difference in the spleen, liver, and bone uptake. There is still a little bit of uptake in the mouse spleen, which was blocked with unlabeled CI-8993 antibody, indicating that CI-8993 shows some cross-reactivity with the murine VISTA antigen. Images obtained with zirconium-labeled IgG1 control do not show high uptake in myeloid-rich organs, and as a result, we see higher blood pool activity. As expected, we did not see any differences in normal tissue distribution between the human VISTA mouse or the wild-type mouse. After imaging was completed, all mice were sacrificed and organs counted for radioactivity. The results of the biodistribution study are shown in the graphs on the right. The graph in the top shows blood and tumor uptake. The graph in the bottom shows the normal organs with highest uptake, spleen, liver, and bone. To evaluate specific tumor targeting, we looked at the tumor-to-blood ratios. Tumor-to-blood ratios indicated specific tumor targeting of zirconium-labeled CI-8993 in the presence of unlabeled CI-8993. After completion of the imaging study, we also ran 2 different assays to confirm the presence of the human VISTA target in different tissues. Our first experiment was a FACS assay in which we used different myeloid markers and T-cell markers to identify the presence of VISTA in these different cell compartments within our tissues of interest. Our data shows that human VISTA knock-in mice expressed human VISTA in the expected tissues, namely in spleen cells and on myeloid cells and some T cells infiltrating in the tumor. FACS data also showed that although in the liver the myeloid cells that are present do express human VISTA, overall the percentage of this cell population is very low. Therefore, we concluded that the zirconium activity seen in the liver was mainly due to normal antibody catabolism and clearance of antibodies. Spleen, liver, and tumor of the human VISTA knock-in and wild-type mice used in the imaging experiment were also used for Western blot analysis with a human-specific anti-VISTA antibody. We used recombinant human VISTA antigen as a positive loading control. We confirmed high expression of human VISTA in the spleen of human VISTA knock-in mice and also some expression in the tumor samples collected from the human VISTA knock-in mice. As expected, No human VISTA was observed in any of the tissues collected from the wild-type mice. Confirming our results obtained with FACS analysis, livers of both human VISTA mice and wild-type mice did not show expression of human VISTA. To evaluate the specific targeting of CI-8993 in a VISTA-positive human tumor model, we performed a second combined PET imaging and biodistribution study. In this study, tumor and tissue distribution of zirconium-labeled CI-8993 was evaluated in a human VISTA-positive human pancreas cancer model, CAPAN2, in biopsy nude mice. We evaluated the tissue distribution at 3 protein dose levels: 1 mg per kilo trace dose, 1 mg per kilo trace dose plus 10 mg per kilo competing unlabeled CI-8993, and 1 mg per kilo trace dose plus 30 mg unlabeled CI-8993. Data is shown of imaging and biodistribution results obtained on day 7 post-injection. At the lowest dose level, highest uptake was seen in spleen and tumor. Increasing levels of competing unlabeled CI-8993 reduced the uptake in spleen and tumor, suggesting the specific targeting of CI-8993 in spleen and tumor in this mouse model. After completion of the imaging study, VISTA expression in CAPAN2 tumors used for imaging was confirmed via Western blot. As controls, we used K562 negative control cells and compared this to the human VISTA expression in CAPAN2 cells. Lysates from CAPAN2 tumors imaged at different dose levels of CI-8993 confirmed the expression of VISTA target in CAPAN2 tumors. Lysate of a U87MG glioblastoma tumor was used as a negative control. In conclusion, we showed that the radiolabeling of CI-8993 was successfully established. Quality control demonstrated that human VISTA binding activity was maintained after radiolabeling, and specific tumor targeting of zirconium-labeled CI-8993 was shown in a human VISTA knock-in mouse model as well as in a CAPAN2 xenograft model. Our results demonstrate that zirconium-labeled CI-8993 is now suitable for use in the planned human bioimaging clinical trial study. With that, I would like to thank everybody who worked on this study from the tumor targeting lab, especially Christian and Alex, who did the radiolabeling of CI-8993. I would also like to thank our collaborators at Curis for this very exciting collaboration project, and thank you for listening.